Hello biologists, we are going to do lab 3.04 on digestion enzymes. Let's talk briefly about digestion first. Food enters through your oral cavity, you chew it up, that's mechanical digestion, that breaking food into smaller pieces. After food is mechanically digested, your saliva starts to chemically digest it by adding amylase, which is an enzyme. Enzymes are molecules that help reactions happen more quickly. Then it travels down your esophagus into your stomach. More enzymes are added in your stomach. It travels through your small intestine right here, which is also called the ileum. That's a long journey, about 27 feet. And then it travels into your large intestine and out it goes. Enzymes are added throughout this process, and they're very important in helping your body break down the large molecules into smaller, more usable molecules. And enzymes are added here in your saliva. They're added in your stomach. They're added by your pancreas right here, this little green thing kind of behind your stomach. The liver, the gallbladder, which stores bile. Those all dump enzymes into the digestive process. The three important functions of the digestive system are to break down food into usable molecules, break the big molecules down into smaller ones, absorb molecules into the blood and move those molecules throughout the body and eliminate waste. So some of your food you can't use, you got to get rid of it. Enzymes work in this process is they take the big molecules and they break them down into smaller molecules. So some enzymes help build molecules, but digestive enzymes are mostly taking the larger molecules, like this polypeptide chain, and turning them into smaller molecules. And your body can handle these smaller molecules and absorb the smaller molecules more easily. So we talked about two types of digestion. There can be mechanical digestion, where you're chewing your food, you're breaking those pieces down, you're breaking like that big piece of chocolate bar into smaller pieces of chocolate bar. And then there's chemical digestion, which happens at a molecular level, where the big molecules are broken down into smaller molecules. Here's an example of a large molecule that we're going to talk about in the lab today, and it's broken down through an enzyme into two smaller molecules. So a disaccharide is broken into two monosaccharides. And that brings up lactase. Lactase is an enzyme that works in your digestive system, in your small intestine, to break down lactose, which is a sugar found in milk. If you look in the back of your milk, it contains sugar. It doesn't contain sugar because they add sugar. It contains lactose, which is a milk sugar that comes right out of the cow. Lactase is an enzyme that works on the milk sugar, lactose. So the enzyme is the ACE, and it works on the sugar, which is the OS. This enzyme works like a molecular scissors, and it chops up lactose, which is a disaccharide. Di means two, so that's two sugars here, into two monosaccharides. So here's the disaccharide. Notice the two rings, and if it's acted on by lactase, this lactose, this disaccharide, breaks apart into two separate sugars, two monosaccharides. Lactase acts like a molecular scissors and snips the enzyme in half right here to make two monosaccharides. So let's talk about the lab, 3.04, 3.06. In the first part of the lab, you need to fill out Table 1. You need to predict if glucose will be present in a mixture of milk and water, milk and hydrochloric acid, milk and lactase, and milk lactase, and hydrochloric acid. So glucose is the sugar. This is the sugar after lactose is cut in half. So if we're just dealing with regular milk and some water, we have to ask ourselves, is glucose going to be present, or is it going to be the disaccharide lactose? If we add some hydrochloric acid, is that going to break up the sugar? If we add some lactase, which is the enzyme that acts on lactose, that sugar, is that going to do anything? Is that going to cleave that lactose molecule, that disaccharide that we've been talking about? Is it going to be able to break that in half? If we add some lactase, will it be able to break it up? And what about if we do that in hydrochloric acid? So you write down your predictions, and I will explain a little bit more about why hydrochloric acid might be a good idea. And this is what the virtual lab looks like. We'll talk about the results for a second here. The stomach is a really acidic place here. And I want to give you some information before you make your prediction. The stomach here on the pH scale is very acidic. It's down by lemon juice and vinegar. In fact, you should probably wear your safety goggles if you're going to vomit, because that acid that comes out of your stomach is pretty nasty stuff. You don't want to definitely get it in your eyes. If you've ever gotten it up your nose or in the back of your throat, you know it's nasty. On the other end of the pH scale are things like soap and drain cleaner. They're the opposite of acids. They're called bases. Now, enzymes, which are big molecules, big, carefully constructed molecules that have lots of interconnections between the amino acids. Lactase is a big, complex molecule. This is the enzyme that breaks down lactose, that disaccharide. Big, complex molecules are made up of a lot of amino acids. We learned that in Unit 1. And those amino acids make interconnections between their side chains. And that controls the shape of this protein. Remember, enzymes are a type of protein, and they make connections between their amino acids. Disulfide bonds are one of the examples of those connections. And so, as the molecule makes connections between the amino acids, this long chain takes on a particular shape, and that shape is very important. It's also very sensitive to pH. So enzymes in your body have to function in particular pHs. And since lactase is found in your small intestine, it has to be able to work at the pH of your small intestine, which is about 6 where lactase works. Hopefully that information on lactase and pH is going to be helpful in making your prediction. Remember, hydrochloric acid will make this milk lactase mixture quite acidic. So we set up the results, we set up the lab in the virtual lab, and we can look at the results. You're supposed to let it sit for 48 hours, but fortunately I've already done that, and I've actually done the experiment in real life, so I can show you in real life what the results are. So in order to figure out whether there's glucose, in milk, we know there's lactose. I have these little detectors. They're actually meant for detecting glucose in diabetic urine, but we'll use them on milk today. And I have a tube of just milk and water here, and we will look for glucose by dipping the strip in the milk and water and seeing if it changes color. Here is just the plain old strip dipped in water. This is the color of just the strip without any glucose in it, and will turn dark brown if there's glucose present, or some shade of dark brown if there's any glucose present. We'll let this sit for 30 seconds, and in the meantime, we'll test some milk that I've added lactate to. Lactate is a commercial product that contains lactase. It's for lactose intolerant people. Later in life, a lot of humans quit producing lactase, that enzyme that breaks up milk sugar lactose, and so they need some help doing it, and they have to take in lactase from another source. You might know someone who's lactose intolerant. So if we add that enzyme to milk, something might happen. So here's our tube of milk and lactate. You can see that the strip's already starting to turn a little bit brown as compared to the one that we put in just plain milk that's still really blue. And you can see that strip turning brown, showing us there's glucose present when you add lactase to milk. It does break up that sugar. So we can write in our test strip results, blue, no glucose, brown, definitely glucose present. Before we look at the results of the milk plus lactase plus hydrochloric acid, let's look at some pH paper here. I actually dipped this pH paper in the milk plus I used vinegar reaction here. And we can see that the pH, if you look carefully, is about 6. Let's test that against the pH of just plain milk. We'll let the color develop there a little bit. Milk's going to be much more neutral on the pH scale. For this one, I did this off camera, and there it's blue. There's no glucose. Hydrochloric acid doesn't break up lactose, so there's no glucose here. And this one turned a little brown. For comparison, this is the one with lactase without the acid, and this is the one with vinegar added. And it turns out we didn't quite get the enzyme, the conditions it's most functional in. So if there's no glucose, there's no digestion. No digestion here. We still got lactose. There's no glucose. Just lactose here. And here we have glucose. Lactose was broken down by lactase. In this case, that was from the commercial brand Lactaid. And there was a little glucose. There wasn't as much as there was in just plain milk plus lactase. I didn't quite get the pH right. Lactase, it turns out, works best at about pH 6. And that makes sense. It doesn't work well at pH 2, which is your